Hi, my name is Victoria Lopez, and this is my presentation on the development of a new therapeutic treatment for ALS. Amyotrophic lateral sclerosis is also known as Lou Gehrig's disease after the famous baseball player's very public battle with ALS in the late 1930s and 40s that led to widespread knowledge of the disease. ALS is a neurodegenerative disease with an average life expectancy of 2 to 5 years after diagnosis that affects about 5 in every 100,000 people. It creates progressive wasting and atrophy in all skeletal muscles via nervous system inflammation, myelin sheath destruction, and motor neuron death, eventually affecting the diaphragm, leading to respiratory failure in the absence of medical intervention such as ventilator support. ALS doesn't have major cognitive impacts, with most patients maintaining all cognitive faculties. This is exemplified in the famous physicist Stephen Hawking, pictured at the bottom right at the early and late stages of ALS, who, despite his years-long battle with this disease, was able to continue making significant contributions to cosmology and physics. While the mechanisms of the disease are well known, the development and onset still eludes researchers and specialists. Research has found genetic mutations linked to the disease etiology, and it's now being recognized as a possible autoimmune disease. Autoimmunity is the body attacking its own healthy cells and tissues. This response is possibly connected to the gut dysbiosis hypothesis, which posits that imbalances in gut microbiota create signals and damage, leading to dysfunction in the nervous system. Researchers are also exploring underlying cellular mechanisms, including abnormal protein accumulation, oxidative stress, and mitochondrial dysfunction, which are thought to lead to the death of motor neurons. The treatments discussed here aim to address the links between the gut-brain axis and development of ALS. The gut-brain axis, or GBA, is a bidirectional line of communication between the gut and nervous system that has very recently been linked to many well-known human diseases. ALS patients have been consistently found to have some form of oral or gut microbiota imbalance, otherwise known as gut dysbiosis. This image illustrates the GBA and the potential connections between gut dysbiosis and ALS pathology, showing the signaling pathways between the gut, immune system, and the central nervous system. We can see decrease in barrier integrity in both the gut and blood-brain barriers, leading to inflammatory responses and mitochondrial dysfunction in the nervous system that possibly marks the onset of the disease. The goal of the treatments being researched now is to improve the signaling within the GBA of ALS patients by healing the underlying dysbiosis. So, how can this be achieved? Research for treating gut dysbiosis in ALS patients is exploring the use of pre, pro, and postbiotics. Prebiotics promote the growth of beneficial gut microbes by providing substrates that support their growth. Probiotics repopulate the gut with live beneficial microbes, and postbiotics are metabolic byproducts of gut microbiota found to have neuroprotective mechanisms. These biotic treatments aim to halt the progression of symptoms that lead to the loss of movement and independence in patients by improving communication signals within the GBA. Currently, much of this research is in preliminary stages and is limited to animal studies, though there have been recent human trials that have shown safety and efficacy with probiotic therapies. In the research of prebiotic treatments, compounds such as fructo and galacto-oligosaccharides have been shown to weaken neuroinflammatory responses by increasing populations of healthy gut microbiota. Though results are still mixed and some outcomes with other compounds have shown no positive impacts. Probiotic treatments with species such as Bifidobacterium breve, Lactobacillus plantarum, and Lacticacebacillus rhamnosus have been successful in reestablishing dysregulated gut populations, leading to delayed neurodegeneration and paralysis prevention. Treatment with probiotics has the most consistently positive results. Postbiotic treatments include suspensions of natural bacterial byproducts, such as butyrate and phenylbutyrate, shown to decrease microglia in the spinal cord, whose overactivation creates excess inflammation. Postbiotics have also been found to block certain factors that lead to motor neuron death. Similarly, results of postbiotic treatment research needs to be continuously honed, as inconsistencies in efficacy have been found. Larger scale human studies need to be conducted to determine the long term effectiveness and safety of these new formulations before they can be made available. If studies are successful in finding an effective ALS therapy with these microbiological substrates, they might look like some of the examples shown below, either in the form of fermented foods, suspensions, or symbiotic pills. Successfully integrating these treatments involves training for hospital staff and caregivers and straightforward administration. This should be relatively simple since training would be drawing on basic skills of patient care, such as administering pills, lab developed suspensions, suppositories, and foods. New hospital implements would include equipment for the preservation of these foods, suspensions or pills, such as freezers and refrigerators, which would be relatively easy additions to most clinical settings. Individual outcomes and patient monitoring protocols would need to be put into place, and there are still questions on how to go about this, such as whether fecal or oral samples are the best route for tracking possible changes in patient gut microbiome populations. Challenges include the aforementioned inconsistencies in treatment outcomes, as well as the diversity and variability of gut microbiota across populations and even between individuals, making it hard to know which microorganisms to focus on in the development of a treatment. At this point in the research, substrates that can be successfully globalized still need to be identified. It's important to note that these treatments aren't cures and instead aim to reduce progression of disease symptoms in ALS patients that would lead to a longer life expectancy. Therefore, these treatments should be made available to patients in the early onset of the disease when possible to maintain as much of a person's motor function for as long as possible. It's really inspiring to see how our increasing understanding of microorganisms is being used to help treat such a devastating condition, and I hope to see the implementation of treatments such as these in my lifetime. Finally, here's my list of references for this research project. Thank you for listening to this presentation, and I hope you learned something new.